RNA will come, this RNA is known as RNA primer. Okay. Later on, what will happen? RNA is not required in DNA. Later on, some other enzyme will come, and that stretch of RNA will be removed. Only DNA will be there. To make one fragment, first of all, one RNA primer will be there, and then DNA. To make another strand, small fragment of RNA will come first, and then DNA will be polymerized. Similarly, if third strand has to be made, small fragment of RNA will come, that is RNA primer will come, and DNA will be synthesized, polymerized. So later on, these RNA primer will be removed. Otherwise, there will be problem if they persist, if they remain there. DNA will not be exist. Okay. So again, let me show you one thing. DNA polymerize this fragment up to this point, and it again goes to this side. It polymerizes from this point to this point. Again, it goes to DNA polymerase goes to this point. It polymerizes from here to here, and these are the fragments formed. And the process will continuously occur till the end. So at last, what will happen? Let me draw again. This is the old strand, template strand. Okay. The process is continuously. Suppose bond has been broken. Okay. And this is the continuous synthesis. Here, new DNA is formed, whose polarity is 3' here, here will be 5', and here will be 5', here will be 3'. And for the another strand, this end is 5' end, this end is 3' end. The case is quite different here. Here are the fragments. This is the end point. Now these fragments has to be joined as there is gap. Otherwise, this is not DNA, no. You know, this is the 3' end, this is the 5' end, this is 3' end, this is 5', 3, 5, 3, 5. So these are the fragments created. Okay. Now, actually, at the beginning, we had one DNA. So at last, we are having 2 copies of DNA. What I am going to explain is that how this gap is fulfilled. Later on, another enzyme, there is another enzyme whose name is DNA ligase. That DNA ligase is like a gum that will come and join this gap. Actually, this is the big gap what I am saying is showing. Gap will not be like this. It will be very small gap. So DNA ligase will come and join. So here DNA polymerase enzyme again works to make hydrogen bond here, and at last another DNA strand is formed. Okay, so now after joining these fragments, Okazaki fragments, both the strands look same. This is continuous, this is discontinuous synthesis. Both of them look same. So there will be no problem. Now, where we came at the end, initially we had one DNA, isn't it? One DNA. That means in one DNA there will be 2 strands. This is one DNA. And at last we have two DNA. This is one DNA molecule and this another DNA molecule. And these DNA molecule, where they are actually located, they are in the chromosome. So one chromosome is divided into two chromosomes, and while dividing this chromosome, this DNA will move to their respective cell. So initially I have shown here two cells from one cell. Only two chromosomes were there, and at last 4 chromosomes formed. Okay, so DNA replication is occurring in this manner. And here what we see, please notice that in the new DNA molecule, so this is one new DNA molecule, this is another new DNA molecule. In this, one of the strand remain old and another strand is new. Here also this is new strand and this is the old strand. Old is known as template, new is new strand. This is template strand, older one, and the new one is the here. So in one DNA molecule there will be one new strand and another old strand. Okay. Student, please note that I tried a lot. This is the first time. I think many loopholes are there. Please understand and go through these paragraphs. And here in page number 106, there are some important points. Here from this point furthermore up to ATP, please note that this is very important point, and you need not skip anything. Please refer all these things and you'll understand easily. So if you have any query, please ask me through WhatsApp messages. Thank you.